Hello everyone, back to my channel with another interesting topic. So today I will be discussing on gene editing technique or gene editing tool used in molecular biology. Before I begin my chapter, I want to introduce you with few of the common terms. Okay? Yes. Let us know about enzymes. What are enzymes? See, there is enzyme means all enzymes are protein molecules, but all proteins may not be enzyme. So, these are the basics that you have to learn. So, let me repeat, all enzymes are protein molecules, but all proteins may not be an enzyme. Okay. Now let us learn the term known as genome. What is a genome? Genome means the complete set of genes or genetic material present in the cell is known as a genome. So these are the common terms which I am introducing you in order to understand the gene editing process technique. So let us understand the meaning of SNP. What is a SNP? SNP means a single base change, termed as SNPs. So SNP means a single base change. Okay, these are the common terms. So once again let me repeat. All enzymes are proteins, but all proteins may not be an enzyme. Okay, with this introduction, I am going to talk about my topic, that is on gene editing technique. Okay, yes. Come, let's begin our chapter with gene editing process. Okay, this gene editing process, the main enzyme which is involved in gene editing process are Cas13 and Cas9 proteins. So if I refer to proteins, that means these are enzymes also. So all enzymes are proteins. So whenever I say an enzyme, it is a protein. Okay, for that reason I introduced the basic terms to you. Okay, the enzyme Cas13 was discovered in the year 2015 and it is a part of the family of proteins that includes Cas9 also, which is an enzyme best known for its role in gene editing processes. So I am introducing with two of the enzymes, that is Cas13 and Cas9, which are protein molecules. In turn, they are enzymes which are involved, which play a very major role in gene editing processes. Okay. In nature, Cas proteins are a key part of bacterial immune systems. So they constitute the bacterial immune systems. These Cas proteins. Okay. They usually what they do, they disable the viruses that invade the bacteria by snipping the invaders. That is, it may be DNA or RNA. That is the role of this Cas proteins, which are present in bacteria. They are the origin. Okay. So what they do, they disable the viruses that infect the bacteria by snipping the invaders, that may be DNA or RNA. So let us know the role of these Cas9 and Cas13 proteins. What are their role? So Cas9 is just like a scalpel which acts as a knife. Okay, the role of Cas9 is like a scalpel. It helps in cutting the required targeted sequence in the gene. So it acts as a scalpel. With Cas9 acts as a scalpel. Cas13 is more like a machine. Okay, Cas9 acts as a scalpel and Cas13 is like a machine. It cuts, they both together, what they do, they help in identifying a specific targeted locations in the gene, and also they slashes off the target gene in the DNA molecule. Okay, that is the role of this Cas9 and Cas13 proteins. So Cas13, which is also referred to as a machine, has already become a very powerful diagnostic tool in gene editing processes. So researchers can use it to quickly identify the viruses in patients' blood, even those present in a very low levels. So these are very specific and they can identify the target. I mean, they can identify this one target molecules even when they are present in a very low levels. Okay, yes. Now let us understand about CRISPR, CRISPR. So CRISPR-Cas proteins together, CRISPR-Cas9 DNA snipping. So that is the tool which is used for gene editing processes. So let us learn about this CRISPR, CRISPR. Okay, it is a gene editing tool of genetic engineering technique in a molecular biology by which the genomes of the living organisms may be modified. So as I already told, genome means the complete set of genes or the genetic material present in a cell. So by using this CRISPR-Cas9 DNA snipping, what we can do, what scientists can do is that they help in the altering the genomes of the living organisms. They can modify the genomes of the living organisms. It is also used in the treatment of many inherited genetic diseases as well as for somatic mutations such as cancer therapies. So these are the things. Now let us come to therefore this CRISPR-Cas9 genome editing technology, it has played a very significant role in gene editing technology. It is gene editing process. So let us see how this works. That is CRISPR-Cas9 proteins. How they work, let us see. It is a unique technology that enables geneticists and medical researchers to edit the part of the genome by removing, adding or altering the sections of the DNA sequences. So how it works? It works by editing by either by adding, removing or altering the sections of the DNA sequences. So it is currently the simplest, more versatile and precise method used for gene manipulation processes, and therefore it is causing a buzz in the science world. Okay, yes, let us see the mode of action, how this CRISPR-Cas gene editing, how it works and things like that. So researchers create a small piece of RNA along with the short guide sequences. This acts as a guide for excision of the target DNA molecule required in the gene. Okay. This in turn, RNA molecule, this in turn attaches or binds to the target sequence of the DNA of the genome, and it also binds to the Cas9 protein. So it plays a very major role. What these researchers, they use a small piece of RNA with a short guide sequences, so that acts as a starting point for excision process of the required genome in the DNA. So what this short sequences of RNA along with the short guide sequence, what they do? They identify both the target sequence in the DNA in the genome of the DNA and also to the Cas9 protein. So but this is the role of this RNA molecule. Like this, it can be used for treating many human diseases, including inborn errors of metabolism. Okay. Now, recently, 2 women won the Nobel Prize for Chemistry for this gene editing process recently. So, they have won. Their name are Carpentier and Jennifer A. Doudna. So, they won the Nobel Prize for this gene editing technology, gene editing tool, that is CRISPR-Cas gene proteins. Okay, these 2 women, they won the Nobel Prize in Chemistry for gene editing processes recently. So, this gene editing technique is also known as CRISPR-Cas9 DNA snipping. They act as a scissors, okay, in order to cut the target DNA required in the genome of the organism. So, this can change the DNA of animals, plants, microorganisms with extremely high precision. So that is why this CRISPR-Cas gene proteins, they have made a very significant role because they are extremely highly, they identify with a very high precision. Means they are very specifically, they can identify the target molecules. So therefore they have contributed a very major role in gene editing technology. So it can be used for cancer therapies also. It has a big dream of curing many of the inherited diseases. So we are hoping to have the cure for many of the inherited diseases using this gene editing technology, that CRISPR-Cas gene. Okay. So as I told, Carpentier and Jennifer A. Doudna, they won the Nobel Prize for gene editing technology. Carpentier discovered a previously unknown molecule which was a part of the bacterial ancient immune system that disarms the viruses by snipping off parts of their DNA. So this was the Carpentier discovered the previously unknown molecule which was present in the bacterial genome that was identified, which was a part of bacterial ancient immune system which disarms the viruses by snipping off. Snipping means removal of one nucleotide. Just change of the, which results in the changing of the amino acid. So viruses by snipping off parts of their DNA. So this was the discovery. Okay. Carpentier worked with Doudna to recreate the bacteria's genetic scissors. So they made the genetic scissors simplified. They simplified the genetic scissors. How they did? They simplifying the tool to be used easily for gene editing processes. So Carpentier worked along with Doudna to recreate the bacteria's genetic scissors, thus simplifying the tool of gene editing process to be used easily for this gene editing processes. So they reprogrammed the scissors to cut any DNA molecule at a predetermined site, paving the way for scientists to rewrite the code of the life. So scientists can rewrite the code of the life also by using this gene editing process, that is CRISPR-Cas9 DNA snipping. So this is all about gene editing process. Hope this video was useful for many of you. Okay then, see you soon with more interesting topics. Till then, take care. Bye-bye.